Okay, so last week we covered ligations and assembly. Ligations in general are combining DNA fragments. If you bring that to the bigger scale, though, it's called assembly, where you start making composite parts that are full plasmids with different, like a prefix part and a suffix part. And to do this, you can either do standard assembly, which is when the prefix part is put into the suffix part backbone, or you can do 3A assembly, where you have a prefix part separate and a suffix part separate, and you put it into an external backbone, which you'll be using most of the time in all of your procedures in lab. The protocol for doing this, at least the 3A assembly, is to gather your cut DNA parts. They're going to be linearized at that point, and your enzymes that you're going to be using for each of the parts. You can find which enzyme you need for each part in regards to last lectures and last videos, and also just on the iGEM webpage where it covers 3A assembly. And next, you add all the ingredients to a single epitube for each part, right? Or to one epitube, because you're combining the parts. You put it in the thermocycler on the iGEM ligation protocol. It'll take a little bit of time, and after that, you store it in the freezer for later use. Or you can use it right then if you have a protocol that you're doing afterwards. I included the little diagram from last time if you want to revisit it again. Just know that the ligation occurs with the covalent bonding, and the hydrogen bonding is when they just match up and they start touching each other. Okay, so PCR is also called polymerase chain reaction. You can kind of figure out what it is based on its name. Poly means many, erase is like enzyme, chain reaction, it keeps on happening, right? And pretty much what this is just linearized DNA copying. Unlike using E. coli to replicate plasmids, this is strictly focused on replicating linearized DNA. So the E. coli can't just like replicate the linearized DNA. It has to be in a plasmid form. So when it starts doing mitosis and all of that, it replicates the plasmid. Here, though, we don't want to replicate the plasmid. We just want to replicate, you know, a promoter, a certain part, a gene coding sequence, whatever. And this happens in 3 steps, which forms a cycle. It starts with denaturation, where the DNA, your part of interest, is being denatured. Then it goes to annealing. I'll go into that, and finally extension. And because it's cyclical, you can repeat this 21 to 30, even like 40 times. And what this does, it creates more and more and more of your part of interest, just copies it again and again. And this is at an exponential rate. It copies, each one of those copies doubles. Yeah. Okay, so the first step is denaturation. And it starts with your part of interest. So if you remember, these are called different things in regards to whichever protocol you're using. But in this protocol, this is called the sense strand, and this is called the antisense strand. It could also be like the coding strand or the template strand, depending on, you know, if you're going over transcription or translation. And in the denaturation protocol, your part is separated, so the hydrogen bonds are broken between each of the nucleotides, and it's separated into 2 single-stranded segments. And this is done at a high temp around 90 degrees Celsius, even higher than that. And that's because at that temp, these hydrogen bonds are going to become really weak due to the kinematics of the solution, because they're vibrating like crazy. And the hydrogen bonds are going to break, and they're going to start separating. Note, though, it's not hot enough for the covalent bonds to break, because the enthalpies of formation are really, really high in regards to the phosphodiester backbone. So it requires a lot of energy, a lot of heat in order to get those broken, and it's not hot enough to do that. So yeah, the backbone remains when you separate these. It's just the hydrogen bonds that break. So why do we want to separate it, though? And that's because in order to make a copy, we're going to need to use these as like template strands in order to replicate them. So each of these can turn into another duplicate strand, which begins in the next step, which is called annealing. And this is where something called primers start coming into practice. I haven't really covered primers at all within these lab committee meetings yet, but pretty much they're little RNA segments that combine or attach to complementary DNA strands. And here, so you can see that the DNA is segmented, your sense strand and antisense strand, 5', 3', 5', 3', like typical DNA. And your annealing takes place at 55 degrees Celsius, where the reverse primer, right here, attaches to the stop codon of the sense strand. So this is the start codon would be right here, and the stop codon. Your reverse primer you make yourself, and it's complementary to that stop codon. So it attaches to the end right there. Likewise with the forward primer, except it attaches to the start codon on the antisense strand. And the reason why these primers attach is so you can begin elongating the DNA so you can form a full duplicate. Without these primers, there'd be nothing for the enzymes to attach to here in order to add nucleotides to the end. Note though, if your reverse primer and your forward primer are of complementary base pairs, meaning like this is A, this is T, C, G, etc., they match each other, they can combine to form a dimer, and you don't want that. If they form a dimer, they can't combine to your template, to your antisense and sense strands in order to elongate them. So keep that in mind. Just make sure that your reverse primer and your forward primer aren't complementary, and you should be good there. You'll be doing more of this within the mini project during the site-directed, or rather the error-prone PCR mutagenesis part of that. You'll have to choose your own primers there. I'll help you with that. You just have to make sure that your primers match with the start codon and the stop codon in your template, in your part of interest. So at the end of this step, you have your sense strand with the reverse primer, and you have the antisense strand with the forward primer. A good way to remember this is 5' to 3' is always, downstream. It's the right way of reading it, the 5' to 3'. And so because this elongates this way, it's reversed, it's backwards. And likewise for this, so it's forward because it's being read from the left to right, from the 5' to 3' direction. The final step is extension, where our enzymes finally become a part of this procedure, and they start attaching to these primers and elongating them. Our enzyme of interest is Taq DNA polymerase, and it binds to these primers and one by one attaches the complementary nucleotides to the ends, and it stops down here where the stop codon is, or where it runs off the end. This does not happen at really high temps because we want those hydrogen bonds to form in this case. So it's going to be right around 72 degrees Celsius. That's enough for the Taq DNA polymerase to be efficient when working with this, but not high enough to start breaking the hydrogen bonds that you're trying to form. Of note though, so because this primer is complementary, while this strand is 3', this segment right here starts at the 5' and this is the 3'. So when this elongates, it's always going to go in the 5' to 3' direction down to form a full complement. Likewise with here, this is the 5', the 3', and it goes down until it finalizes. Right here, it adds the nucleotides, but it doesn't just do that. It also forms the phosphodiester backbone. So it knocks off, I think, like a phosphate group, and it attaches the nucleotide to extend the phosphodiester backbone. Without that, it would just be a bunch of nucleotides hanging on by hydrogen bonds, which is not structurally sound. And that's pretty much it. After this finalizes, the Taq DNA polymerase has completely replicated your part of interest and has created 2 duplicates. And afterwards, you can start the procedure again, hit it up to a high temp, 90 degrees Celsius. It will break the hydrogen bonds and do the procedure all over again. You can do this 20, 30 times, and you'll have thousands upon thousands of replications of the same DNA part. I wanted to include a few PCR applications. This is by all means not all of them. There are plenty of others, but these are the main ones. So what directly relates to your mini project are the error-prone PCR. And what this is you just introduce a mutagen. I think it's manganese. You add it to the mix, and it starts making the DNA, Taq DNA polymerase, makes it error-prone, so it might switch a nucleotide here and there, might miss one, and that in turn causes mutations within your DNA part when it does the replications. Next is just creating many copies of the same part. That's the general purpose of this. If you want to have enough concentration of your part, you would use PCR. You could also use cloning. You could just put your part into a plasmid and replicate it in E. coli. It's less efficient though, and it's harder to do. This is faster, and it's also more efficient. And finally, Sanger sequencing uses PCR techniques. And what this does is in order to elongate those parts, it has to use certain nucleotides, and your spectrometers, your computer recognizes that certain amounts of nucleotides are disappearing from the solution and being added to the backbone. And so they can use that in order to sequence the DNA strand, figure out what nucleotides go where. Okay, so the protocol now, it's not that bad. Here's a little diagram in regards to the different ingredients you'll be using. The ones in square are in the master mix we already have. That's the Taq polymerase, or enzyme, the buffer for the polymerase, and also the nucleotides. It's like dt-DNAs or something like that. It's the precursors to the nucleotides that you'll be using. It's already in the master mix. And so, so you add water along with your Taq master mix, your template DNA, so your DNA sample, your primers, and you add all that to a PCR tube. You can mix by pipetting. You can be a little bit rough, once again. Just make sure that you're being careful with the enzymes, keep them on ice, etc., etc. You just mix up and down a few times. After that, you spin it down in the mini centrifuge just to get it all mixed up. And you throw it in the thermocycler. And after that, it's pretty much done. You put it on the iGEM PCR protocol, wait a few hours. I don't know how long it typically takes, I forgot. And after that, you take it out, store it in the freezer, or you can use it immediately if you want. Yeah, pretty simple. Okay, next time I want to go over the sequential mini project steps from the very beginning stages of just planning it, all the way through the different procedures you'll be using in order to outline it so you guys have a good understanding of what to do when you start going into lab and start doing it by yourself. Just know that you don't have to be fully competent with regards to the mini project before you're doing it. The main goal of the mini project is to expand your learning potential, to learn throughout the procedure, to learn techniques of how to do things, and just get familiar with the procedures in general. Also, I've narrowed down a few topics that I'm interested in. I don't know if you guys want to vote on this or anything like that, but the ones out of the ones, the topics of the ones I mentioned that I found most interesting are CRISPR, autoinducer 2 because it relates to our project and getting you guys competent about what we're doing with our project is good, and other types of assemblies like Golden Gate to expand the methods that we could use in order to get our parts working in the future.